Welcome everyone. So we have a bit of a change of pace today, and we're going to talk about more philosophical aspects. So I'm very pleased to present Dr. Emily Parke, who is a lecturer in philosophy at the School of Humanities here in Auckland. And her research interests lie at the intersection of the philosophy of science, biology, and applied ethics. And she's done some past work in synthetic biology and experimental microbial evolution. And so her research focuses on philosophical questions about scientific inquiry and methodology, and the ways in which studying microbial life and probably even simpler organisms shapes our knowledge of life and living things. So hand it over to Emily.
Thanks. Yeah, I thought I might start by saying a little bit about who I am and why I'm here, but that was a great introduction, so maybe I don't need to say much. But I guess the main thing I wanted to say was that often when you hear about philosophy of biology or philosophy of science, I think the first thing that tends to come to mind is bioethics. So ethics is part of what I do and part of what I'm interested in, but most of what I'm interested in is asking sort of conceptual questions about how science works and how scientific knowledge is generated, which was the focus of my PhD thesis and the focus of this talk today. So I'm going to be talking about sort of a mix of ideas from my PhD, which I finished last year, and several papers in progress at the moment, and then I'll say a bit about some future directions at the end, all focused around this question of experiments and models. Okay, so I'm going to start out with an example. In a 2002 paper in Nature, Ben Kerr and colleagues investigated whether biodiversity can be maintained when communities with non-transitive interactions compete only locally. So the key idea of non-transitive interactions is that they're basically like a rock, paper, scissors game. So a simple version would be if you have three species where each species outcompetes one of the others, but neither species outcompetes both of the others. So like in the classic kids game where scissors beats paper, paper beats rock, and rock beats scissors. There's been some previous modeling work on this. What Kerr and colleagues did in this paper is to build a model of non-transitive communities based on the idea that three bacterial strains could instantiate this sort of rock, paper, scissors dynamic. So they set up a model with three strains called C, S, and R, where C would produce a toxin called collicin, S would be sensitive to it, and R would be resistant to it. And furthermore, so C would kill off S, obviously, but S would have a slight competitive advantage over R and R over C. So this sort of three-way rock, paper, scissors dynamic. Here are the simulation results. So their aim was to compare when interactions are only local, as on a lattice, versus when interactions are global and everyone disperses and interacts with everyone else. So in a lattice-based version of the simulation here, where the three strains are represented in red, green, and blue, they showed that biodiversity is maintained, or the three species coexist, as long as their interactions are only local. So they form clumps according to type, but none outcompetes the others. So when they ran the simulation on the lattice, they got these clumps that would sort of chase each other around the grid. When they ran the simulation with everyone interacting with everyone else, on the other hand, when the interactions were global, diversity was lost and one strain would eventually outcompete the others. So these two graphs up here are just showing time horizontally, abundance vertically. So with local interactions only, there's coexistence. With global interactions, one strain drives the other two out. So these results were robust over many runs of the two versions of the simulation. But then to test the results, they went and set up the same sort of community structure in actual bacteria, in E. coli, in the lab. So they made three strains of E. coli, one producing collicin, one sensitive, and one resistant. And the results were essentially the same, except this time the strains were chasing each other around a petri dish instead of a lattice on a computer screen. So this is just showing the same sort of thing that went on in the simulation, where you have clumps of the three kinds of bacteria on a plate. The yellow lines represent the interface between C and S, and the pink lines are the interface between C and R. So the main point was that again, you got clumps, and the three moved around but continued to coexist stably when they were interacting locally. When they ran the same strains in well-mixed flasks, one ended up driving the other out. So these experimental results were taken as validation of the hypothesized and simulated original claim about biodiversity. And the authors go on to speculate that this phenomenon might hold in other natural communities as well, like plants and biofilms. So this is a relatively familiar sort of reasoning that starts with a computer model and goes on to actual bacteria in the lab, ultimately making a claim about the dynamics of non-transitive communities in general. So the kind of claim I'm interested in today doesn't have so much to do with this particular example, but with the logic of that reasoning, which is that most people intuitively tend to want to say that they got closer to learning about the natural world or about biological communities in nature when they studied the E. coli, the second example I gave you, versus the computer model. So one might think so because of specifics of the study or not. Regardless, when people think that, it's at least partially due to a general background belief that experiments tell us about the natural world in a way that models don't, or that when we do an experiment, we're automatically closer to learning about nature than when we study a model. So that's the idea I'm going to talk about today, not in this particular case. My aim isn't to take a particular stand on that, but about this general background belief about the relationship between experiments and models in general. So there's a general feeling that experiments are better than models in the sense that they generate better scientific knowledge or put researchers in a better position to make inferences about nature. And this way of thinking is pretty pervasive, at least in many groups that I've given this talk to. Maybe not in this group, I'll be interested to hear what you think, but in the biology programs that I've worked in the past, it's not uncommon to hear things like interesting results, but it's only a simulation, or nice model, but where's your evidence from the real world? So a number of people, both philosophers and scientists, have put this sort of idea into writing recently. So Francesco Guala, who's a philosopher of science, wrote, "Ever since Galileo and Newton, it has been agreed generally that experiments constitute the ideal, most reliable source of scientific knowledge." Stephen Peck, who's an ecologist, writes, "Simulation's utility is debated, and some ecologists and evolutionary biologists view it with suspicion and even contempt." And Mary Morgan, another philosopher of science, says, "The intuition of both sciences and philosophy is that experiment is a more reliable guide to scientific knowledge than models." So the intuition behind these types of claims typically has to do with the idea that when you do an experiment, you're studying something that's more like the natural world. So the kinds of things in the natural world that we ultimately want to learn about, like human societies or biological communities or planets and galaxies, are really complicated and messy. And the intuition behind thinking that experiments are better typically has to do with the idea that when scientists engage with an experimental object of study like Drosophila or a human subject in the lab, they're engaging with something that's plucked from that same messy, complicated world, and a bunch of details come along for free which make the thing they're studying somehow part of that world. Whereas when they study a computer model, for example, they're engaging with this two-dimensional thing essentially made of mathematics and visualized on a screen, which is a distant abstraction compared to the sorts of things on the left. And when people think this, they typically don't think that that doesn't mean that you can't learn important things from the model, just that it's necessarily less like the thing that you ultimately want to learn about. And therefore you have to work harder to make an inference from the model to the world. So in my experience, many people believe something along the lines of this view about experiments versus models, and that's basically the view that I want to challenge today. So two claims that I focused on in my PhD, two reasons that people think experiments are better. The first says that experimental systems are specimens of the natural world, while models merely represent the natural world. That's a way to kind of develop that intuition I was just talking about. The second says that models can't surprise us the way that experiments can. So for the rest of the talk, I want to say why I think these sorts of views are problematic and why the resulting conclusion about experiments being better is problematic too. I think I'll only have time to talk about the first, but I'm happy to talk about the second in questions. So before moving on to talk about these claims in detail, a slide that I think it's good for philosophers to include in their talk, who cares? So I'm a philosopher talking about a debate that some scientists, but mostly philosophers of science, are actively engaging in. So why should scientists care? I think this project has broader implications for at least two reasons. So first of all, people evaluating science often subscribe to the sort of view I was talking about, that experiments are the best. So I have in mind here funding agencies, journals, other scientists evaluating each other's work. Obviously not everyone thinks this way, but I think the kind of judgments on display in those quotations I read a moment ago are pretty pervasive. So it's not uncommon to hear people expressing the idea that all else being equal, it's always better to do an experiment. Second, for science education, students still have it drilled into their heads in school that there's such a thing as the scientific method with a capital T, and it basically goes, ask a question about nature and plug it into a formula and spit out a result. And that formula usually involves coming up with a hypothesis and then designing a lab experiment to test the hypothesis. So I think before the undergraduate level and sometimes beyond, students learning about science aren't really learning about the extent to which scientists are increasingly doing computational modeling in addition to experiments, or often instead of experiments. So if you Google scientist, all of the images that you come up with look like this. I had to scroll down for a while to find an image chunk that wasn't all white men, but that's a subject for another talk. The point is, when people think of scientists, they tend to think of scientists in the lab in lab coats doing experiments. So I don't think there's enough focus in science education or in public perception of science on the fact that modeling is also legit science alongside experiments, or sometimes instead of experiments. So the philosophical project of challenging these ideas about experiments being better has some practical implications too. Okay, just to be clear what we're comparing here. When people compare experiments to models, they often have in mind mathematical models or computer models. But people also talk about simulations in a broader sense to mean a study of any model. So that would include both computer models and physical models, like model airplanes or laboratory analog models or model organisms. So the people who argued for those three claims I read about why experiments are better typically have in mind experiments versus models and simulations in this broader sense, including physical models. So that's the sense that I'll adopt in responding to them. But my ultimate view here is that there's actually no interesting or important difference between experiments and studies of physical models. It's kind of a controversial view, at least among philosophers, so I'll say more about why I think that later. So one way of fleshing out the intuition I discussed earlier is this idea that models only represent the natural world, while experimental systems are specimens or reproductions of the natural world. And then the idea is that this gives experiments the upper hand as ways to generate valid claims about the world. So by specimens, the people who have argued for this sort of view mean something like typical instances of the entities or phenomena in the world that theories refer to. So Levy and Curry's central example of a specimen is Isaac Newton's famous crucial experiment where he studied beams of light in his laboratory in order to learn about sunlight generally. And the contrast to representations, which is what models do, representations here are thought of as intended stand-ins for a broad range of phenomena which contain significant deviations from accuracy and completeness in standing in for those phenomena. So I'll just mention a more specific version of this same claim, which people have argued for, which says that experimental systems are materially analogous to the world or made of the same stuff as the world, in contrast with models which correspond only formally or abstractly. So Mary Morgan's argued that we're more justified in claiming to learn something about the world from experiments because the world and experiment are made of the same stuff. Basically, they're saying that to be a specimen is to be the same kind of material or physical or biological thing as the ultimate target of inquiry in nature. There are annoying, nitpicky philosophers' reasons why we might want to separate this argument from the other one, but really I think that this is more or less a version of the other claim I was just talking about. So for the purpose of this talk, I'm just going to lump them together and call them the specimen versus representation view. So just summing up those same points again with pictures. If we're interested in learning about how human populations behave, consider the difference between doing an experiment and a simulation. The view says that if we study experimental subjects, like our humans in the lab, the views I discussed want to say that we're manipulating a sample or a specimen of the thing in the world that we're interested in. It's the same kind of thing. Whereas if we study a model instead, like an agent-based model on a computer screen, we don't have the same kind of material correspondence. So the population of dots on a screen is a really distant stand-in for the population of humans in the world, and the correspondence between the two is merely formal. So from this sort of point, people have generalized to the claim that it's better to do experiments whenever possible than study models. And again, these authors intend this point to apply to simulations understood broadly to include not just computer models, but also physical models like model airplanes, model organisms, and so forth. In any case, they want to say that the experimental systems are more like the world. So my way of responding to these claims, I look in more detail at some examples from experimental microbial evolution, which I think challenges a lot of these distinctions that people have wanted to make between experiments and physical models. So I'll just talk through a couple of examples there pretty quickly. So experimental evolution involves propagating organisms in the lab as a way to study evolution in real time. The organisms are often microbes because they're really small and you can keep a lot of them in a small space, and they have fast generation times and are experimentally tractable in a number of other ways. So a famous example of this, which you might have heard of, is Richard Lenski's long-term evolution experiment. So Lenski's lab started with a single genotype of E. coli and seeded 12 initially genetically identical populations in identical environments, just flasks of minimal bacterial growth medium, and started evolving them via serial transfer, which means every day as the populations start to use up the resources in their flask, you transfer a representative sample to a new flask so that they can keep evolving. They started this experiment in February 1988, and it's still going today, so the populations have gone through more than 65,000 generations now. I'll just say a bit about two claims about the world outside the lab that were made in two different papers from Lenski's long-term evolution experiment, and use this as a way to respond to the specimen versus representation point. So in the first case, Symonds and colleagues in a 1997 paper say that their explanation for patterns of evolving mutation rates in the laboratory populations might also explain evolving mutation rates in E. coli and similar bacteria outside the lab. So most new mutations aren't beneficial. If you just think about mutation intuitively, there are a lot more ways to mess something up at random than ways to improve it. So you might think that in well-adapted populations and constant environments, the genomic mutation rate might stay the same or even decrease. But in just 3 of Lenski's 12 populations, they saw the opposite. After several years of the experiment, most of the populations maintained the wild-type mutation rate, and just 3 of the populations evolved a much higher mutation rate, about 1 to 2 orders of magnitude higher. And their explanation for how this happens in the experimental system is that mutator alleles, or alleles which increase the whole genomic mutation rate, arose randomly by mutation and then hitchhiked to higher frequencies, which is possible because the populations are completely asexual. So from here, they go on to make a claim about bacteria in nature. Specifically, they say that their findings and their hypothesis for how high mutation rates evolved in 3 of their 12 lab populations might also help explain observations of mutator alleles in pathogenic E. coli in nature. In a second case, in the second paper, Elena and colleagues saw a pattern of cell size evolving by jumps in the lab populations after 3,000 generations of evolution. Sorry, this is really small, but this is just time horizontally, cell size vertically. So the point is that the cells stayed the same size, then dramatically increased in size, stayed the same again, increased quickly again, and so forth. So they say that these punctuated trends that they saw in the lab populations were associated with the rise of beneficial new mutations that quickly swept to fixation. And in the paper, they say that this short-term evolutionary dynamic in the lab might tell us something about punctuated equilibrium, which is the idea that evolution occurs in long periods of relative stasis followed by short bursts of rapid change. This was a hot topic in the mid-90s in discussions of macroevolutionary trends in the fossil record, notably championed by Gould and Eldridge. But they were talking about hugely longer timescales spanning speciation events. So this paper was met with some criticism for what people thought was an unfounded inference. My point today isn't to criticize it per se, but just to compare these quite different looking two inferences. So one from dynamics of mutation rate in E. coli and similar populations in nature, and another about an arbitrary set of lineages whose traces are left in the fossil record. So the specimen versus representation view tried to draw a categorical line between experiments and simulations, and to say that experiments study specimens, which are like the natural world, in a way that representations aren't. Again, a crude way to put that was to say that experiments in the world are made of the same stuff, whereas simulations aren't. So if we adopt that view and try to evaluate these two different inferences from Lenski's experiment that I talked about populations of E. coli in nature versus long-term dynamics in the fossil record, I think we run into some problems. So first of all, is Lenski's work experimenting or modeling? It's called experimental evolution after all, but some people want to say that all work with model organisms is modeling. And there might be good reasons to think that the mutation rates paper looks more like an experiment, and the punctuated evolution paper looks more like a case of modeling, given how close and distant the relationships to the relevant parts of nature are. But that seems strange, I think, because they come from the exact same lab work. And second, it's not always clear how to even evaluate this idea of specimenhood or material similarity. And that's the more important point here. So especially in the punctuated equilibrium fossil record case, I think this comes across nicely. The claim there is about a long-term macroevolutionary dynamic. And the particular identity of the populations instantiating that dynamic at nature isn't really what's at stake. And neither is there phenotypic or phylogenetic closeness to E. coli. So it makes sense to talk about the E. coli in the lab as a specimen of the natural world in this case, only to the extent that both are biological populations. But that identity simply isn't what's at stake here. And in fact, all of the criticisms targeted at this paper focused on the dynamic itself and how it's problematic to extrapolate from 3,000 generations of clonal evolution to claims about events like the Cambrian explosion. So I think the question of whether the laboratory system and the target populations or entities in nature, the question of whether or not they're made of the same stuff or one's a specimen of the other, stops making sense in cases like this. So the first lesson from thinking about cases like this, summing up the points I just made, is that experimental systems aren't always obvious specimens or made of the same stuff, as Morgan put it, as the systems in the natural world that we use them to draw inferences about. And furthermore, it's not always clear what that even means. What got me started on this whole topic in the first place was working in a lab doing experimental microbial evolution, and hearing people say things like experimental evolution is just a really expensive simulation procedure, or experimental evolution is a funny name because it's more like a model of evolution. Ultimately, I don't really think it's interesting to get hung up on the difference between experimental systems and models, physical models, in cases like this. Because I don't think it ultimately matters what we call them. I think the really interesting question here is, how on earth do we get to make general claims about evolving populations in the world by studying these tiny flasks of asexual microbes in environmental conditions that are quite unlike environmental conditions outside the lab? And what makes those claims sometimes sound quite believable and sometimes sound like a bit of a stretch? So I'm interested in understanding those sorts of questions, not so much on the question of whether these are ultimately should be called experimental systems or physical models. So back to this slide here, I think the same goes for the other examples I talked about of what are typically called physical models, like mouse models, the Miller-Urey experiment, or model airplanes and wind tunnels. So they're all examples of physical things in the lab being manipulated in an attempt to learn about cancer or the origin of life on Earth or aerodynamics more generally. In each case, there are obvious senses in which the thing in the lab is very much like the ultimate target of inquiry in the world, and also very much unlike it. So we could argue about whether these physical models are actually more like specimens or more like representations in a given case, and people do, but I think those arguments are pretty impossible to settle. And in any case, they hang on what they're being used as specimens and representations of. So that's why I don't think there's really an interesting or important difference between experiments and physical modeling. I don't think it matters ultimately which of these things we call the stuff on the lab bench or in the wind tunnel or what have you. What does matter is understanding how we can interact with these sorts of things and draw inferences to the world outside the lab. So that's the issue that I think is really interesting here. In other words, to the extent that there is a difference between specimens and representations, it doesn't track a difference between experiments and models. The cases I discussed from the Lenski experiment show this pretty nicely with respect to experiments versus physical models. But I think this point is true for experiments versus mathematical and computer models as well. So I'll say a bit about why. The classic reason that people make the claim about experiments versus models being like specimens versus representations tends to look something like this. So when we think of a classic case of studying an experimental specimen, we might think of something like studying crows, a small group of crows in the laboratory or the field as a way to learn about crows generally in the world. And we might contrast that with a case of representation, like studying the Lotka-Volterra equations as a way to learn about predator-prey dynamics more generally. Or another case of representation, studying individual base models like Sugarscape, which is a model used in the social sciences to learn about social or economic dynamics in human populations. But studies of laboratory systems can also look more like what people have wanted to call representations and not so much like specimens. So that was the main point of the experimental evolution cases I discussed. So the first example from Lenski's experiment looked more like a specimen, the inference from mutational dynamics in the lab E. coli to mutational dynamics in E. coli period. But contrast that with the second sort of case, which was this inference from short-term evolution of cell size to a claim about punctuated equilibrium. Unless any evolving population counts as a specimen of any other evolving population, this looks a lot more like a representation, a more distant stand-in for a broader class of things, abstracting away plenty of details. Finally, and perhaps more controversially, I want to say that mathematical and computational models can also look more like what people have called specimens, and that it just depends on the context of inquiry. So stepping away from biology examples, there are other fields where individual-based models like Sugarscape or the game of life are studied as instances of evolving systems or systems with emergent dynamics. So I'm thinking about research in artificial life or complex system studies. Sometimes the goal there is to say something about evolving systems and emergent dynamics, not as on display in any particular populations in nature, as we ultimately care about it typically in biology, but just at the abstract level, period. So I think it's right to call these sorts of models in those instances specimens of exactly the class of things that researchers want to learn about in those contexts. So the key point here is that if we must talk about specimens versus representations, I don't think there's any clear line there separating models of any sort from experiments. But in any case, I think the difference between specimens and representations isn't as clear as people want it to be. Especially in biology, where the very nature of the subject matter is variation, it seems strange to talk of perfect specimens or samples in any case. Sure, several crows are representative of all crows, and it would seem strange to say that E. coli is representative of all lineages in the fossil record in this example in the exact same sense. But I think the difference here is a spectrum, or a matter of degree, from resembling all members of a target class in many ways to resembling them in fewer ways. And there will be plenty of cases in between that we could argue about. So for example, cases like studying mice as proxies for humans in biomedical research. If we're interested in some specific homologous structure, then maybe this sort of case looks more like specimens. If we're interested in studying toxicity, then these sorts of cases might look more like representations. So I think it's debatable. So that's one of the broadest lessons I want to draw here. Almost nothing that scientists study is a straightforward specimen in the strict sense that I think people making this claim about experiments being better typically had in mind, in the sense that almost all of science is about studying something to learn about something else. And most of the time, I want to say almost all the time, the things scientists interact with directly, whether it's in the lab or in the field or on paper or on a computer screen, is a representation, if by representation we mean a non-identical stand-in for some broader class of interest in the natural world. So in really rare cases, the thing being studied might be a proper specimen. So for instance, if you're studying a small, really clearly delineated population, and your goal is to say something about exactly that population. I think that happens sometimes in conservation biology or anthropology, for example. But almost always, the thing you're studying is at best a sample, capturing some features and extracting away others, and maybe idealizing others. My point here, though, isn't that we should always be skeptical of what we learn from non-identical stand-ins. There are, of course, plenty of robust ways to justify claims from the thing you're studying to the natural world. Just that we should be skeptical of the claim that studying a physical system in the laboratory or in the field means that you're automatically studying something that's like the rest of the world. Even if it's made of the same stuff, in Morgan's terms, the environmental conditions can vary hugely. So this, together with the previous points, I think undermines the arguments I mentioned at the beginning about experiments being better than models. So the key point is that classifying a case of research as experimenting versus modeling doesn't license any automatic conclusions about the value of what we can learn from it. So back to this question, are experiments better than models? It should be clear by now that the answer I want to give is no. But more importantly, while I thought it was a kind of snappy title for a talk, my main point is that it's not really the right kind of question to be asking. It might be a good question to ask in particular research contexts or classes of situations, and I'll say more about that in a moment. But the debate I'm responding to here asks that question about experiments versus models full stop across science. Remember the quotations I opened with. All of those were claims about the difference between experiments and models in an all-inclusive sense that was meant to generalize to any case of experimenting and modeling. And that's what I want to say is really problematic here. So just to be really clear, here's what I'm not saying. When I say experiments aren't better than models, period, I'm not saying that models are better than experiments, or that experiments and models are equally valuable, or that we should trust models more than we do or trust experiments less than we do. All three of these other sorts of conclusions would likewise be problematically generic claims about experiments versus models across science. So my main point is that we shouldn't make these sorts of claims at all. Okay, so I said, are experiments better than models is the wrong sort of question to be asking. Here's what I think is the right question, the sort of question that we should focus on instead. Are the relevant similarities being captured between the thing being studied, whether it's in the lab or the field or on a computer screen, and the ultimate systems or phenomena or dynamics of interest in nature? This is what I talked about in the last chapter of my PhD and a few papers I'm currently working on in future directions. In addition to just asking this question, I think there's something further to say about classes of situations where it might be better to start with studying a physical system versus a computational or mathematical model. Though again, of course, they're often most profitably studied in tandem. So here's what I have in mind there. This is something I've been thinking about more recently. I've said we can't make principled judgments across science about experiments or models being better. I think developing an account of contexts of inquiry or classes of situations where one has privilege over the other is a good starting point. So which sorts of considerations might be more helpful in judging whether experimental or modeling approaches are better in a given context? So by context, I don't have in mind necessarily things like physics versus biology or microbiology versus biomechanics. There might be things to say about research areas like that. But what I have in mind is more like features of scientific questions that make them more prone for addressing fruitfully with modeling approaches versus experimental approaches. So I think one sort of good starting point for an answer here is something like what philosophers have called how actually versus how possibly questions, where the names are pretty self-explanatory. So how actually questions ask about how things actually are in the world. So a how actually question would be something like, what is the length and sequence of the tardigrade genome? Or how did life emerge from non-living matter on Earth? And that's contrasted with a how possibly question, which asks how things could be in principle. So how could life in principle self-assemble from non-living chemistry, regardless of how it actually assembled on Earth? So how actually and how possibly questions are quite closely connected, but we can certainly ask how possibly questions independently of how actually questions. Another way to put the basic idea and have in mind here would be something like more fine-grained versus more coarse-grained questions. So we might be asking about a fine-grained question where the physical or phylogenetic or physiological particulars of the system in nature really matter, versus a question where we're just interested in some high-level dynamic and we don't care about any particular population that instantiates it. So a more concrete example of the kind of difference I have in mind here is, for example, the difference between a question like, how do I understand interspecies interactions in a particular kind of biofilm, where the details really matter. I'm asking about particular species of bacteria and what they're doing, versus a question about the evolution of cooperation, like how could cooperation ever emerge in a world of egoists? I think this is a great example of an area where the question is what I'm calling very coarse-grained and high level, and there's a tradition of learning a ton from iterated prisoner's dilemma situations capturing the formal structure of the issue, not necessarily from looking at organisms and watching how they cooperate as a starting point. Okay, a related, probably more obvious point, another sort of consideration that matters in judging whether experimental or modeling approaches might be more fruitful is thinking about the trade-off versus realism and control. So for example, realism might matter a lot more if we're asking a question about the effects of a new drug cocktail in the human body. Control might matter and realism not so much for asking a pretty basic chemistry question like what will be the effects on molecular bond angle of swapping one atom for another. We have reliable modeling approaches, and it would seem silly in a case like this to say that you're necessarily going to get a better answer from actually swapping the atom in a molecule in the lab, because you can look at models to answer the question. So, okay, I'll wrap up. That's just a start at getting at the kinds of considerations that I think matter in thinking about the relative payoff of studying laboratory systems versus computer models in a given context. But again, the main point I wanted to make here is that I don't think there's anything to say about experiments or models being better, period. We have to ask these more context and class of question type questions about them. So thank you to my former lab and lots of people who have given me helpful feedback on this, and in particular two people who I'm currently collaborating with papers on the moment about these ideas. Thanks.
Thank you, Emily. So we do have time for questions. So, Peter?
In one sense, asking the question about experiments versus models is like asking, which is better, men or women. Most people would agree you need both. But I wonder whether the refinement is more to do with what type of models you use in terms of, I mean, for a lot of bioengineering models, you build in the laws of conservation. You satisfy thermodynamic constraints, or you build in conservation of mass, so that when you're doing, and that you iterate between experiments and models, but the real value the models bring is because then you can interpret your experimental data in the context of knowing that you're satisfying the conservation laws of nature.
Mm-hmm.
So I suppose I'm still puzzling as to why anyone would ask a question about models versus experiments. And there's lots of classic examples where the iteration between models and experiment. I mean, one good example is in the world of electrophysiology, cardiac electrophysiology. There was a point at which there was a lot of debate around whether a sodium-calcium exchange mechanism, whether its stoichiometry was 1 to 2 or 1 to 3. So 1 calcium to swapping for 2 or 3 sodiums. And the modelling could absolutely clarify that it had to be 1 to 3. And that was a nice example of where the modelling led the experiment, so you could then design the experiment to really sort of check the hypothesis that had come up through the modelling. And just as there's plenty of examples where you've obviously got to do the experiment to understand what parameters go into the models.
Mm-hmm.
So it's the iteration between the two of them that's the really valuable.
Yeah, I think that's really important in taking scientific practice seriously, and I think at least some threads of the philosophical debate here have taken the conversation as if, you know, scientists decide whether to do one or the other, and that decision affects the sort of inferences they can make, but exactly, so much of the actual work isn't one or the other, it's the two happening in tandem. Yeah, so I agree with you.
By complete coincidence, I finished reading a paper at lunch today that's in the latest issue of Oikos.
Mm-hmm.
I can't remember who the first author is, but Miguel, who's written a number of, he's an ecologist, has written a number of papers on this, but their interest was in just talking about distinguishing between good and bad models.
Mm-hmm.
They talked about experiments a bit, but I mean, I too am puzzled at the validity, at the utility of a question like which is better, because you can have a lousy model and you can have a lousy experiment, and you know, so it kind of depends. And sort of some of the work I've been doing recently, we've got plenty of examples of both. But in this paper that I finished reading today, they were talking about how we should evaluate models, and they kept coming back to their ability to predict, given novel sources of data. And another metric that you can use to compare, and it would be the same with an experiment, of course, but another metric would be what they call transferability. I'm not exactly sure that's what you mean by coarse and fine grain, but it kind of seems similar, and that is, you could do an experiment under a very controlled situation, like the Minsky ones, for example, or you could do one in a more natural setting, in a pond, for example, where it's open to the elements, and you know, you're allowing more factors to vary. But ultimately, I think I think it's important to distinguish between laboratory experiments and experiments in the field that can at least attempt to capture unknown parameters. I mean, another paper that sprang to mind is a paper that was written by Paul Dayton, I think back in 73 or 74, where he was talking about intertidal experiments, and he made a series of predictions, carried out the experiments, and his initial model actually gave the right prediction, but for completely the wrong reasons. And the fact that models give correct predictions doesn't mean that they're explaining the right parameters, you know, that their understanding of causation is correct. Ultimately, if you've got both transferability and prediction, you must actually understand it. And so I think the things are sort of iterative, aren't they? The relationship between experiments and models, they sort of inform each other.
Yeah, definitely. And I mean, yeah, you're getting into some of the points that I've been thinking about in the related but separate discussion of surprise and whether models and experiments differ in their ability to surprise. But yeah, I mean, I think, like you said, there can be good, bad models. There can also be good, bad experiments. And I mean, the field versus lab experiment case is interesting because I think there's, as part of these ideas about experiments being more like the world, there's also a tendency to think that field experiments are always more realistic than lab experiments, which might or might not be true because there's a huge degree of manipulation. Some field experiments are almost like observational studies, and others involve huge amounts of imposition of artificial structures. And so, yeah, I think part of my bigger project here is just saying we really can't make generalizations about any of these contexts, and there's, like you said, various ways that they can be better or worse suited for the purpose at hand, or put us in a better or worse position to make predictions. But they ultimately work best together in most contexts. Although there are also contexts where we can't do experiments at all on the subject matter of interest because the thing we want to learn about is too distant in space or time, and those are cases where models are sometimes better just because they're all we can do. So, yeah.
And I think there's also a third player here that is often ignored, that in many ways is the most important, and that is the most new discoveries in science don't start with either an experiment or a model. They start with someone designing a new instrument, a new type of measurement. So I think that, most good science is a three-way iteration between developing instrumentation to make new types of measurement, doing the experiments, and then interpreting the experiments with models, but iterating between all three, because you need to keep improving instrumentation to make better measurements, to resolve the questions that come up.
Yeah. Yeah, thanks.
We've done an interesting combination of bioengineering and biology. I'm a biologist and I hang around this place. And one of the clinicians was doing a study on the treatment of fluoride and whether it was a good a treatment for osteoporosis. And so we got all the data from the patients, and we looked at their micro-CT and their histology and things. So we did all the experimental bits that we knew what to do. And then Justin Fernandez and ABI then tried to model the risk of fracture in osteoporotic models. And he actually, the physician I might add, was incredibly impatient and thought it was a complete and utter waste of time. And I said, just, it's not going to do any harm to just let the ABI guys have a go at it. And in actual fact, he came up with questions that we didn't even ask, and the data was actually in the micro-CT, but we were not using it. So that's an example of how the models and the experiments, and ultimately an observational study of humans, was a benefit. And that has actually led into being able, because there's a debate, particularly in the States at the moment, of whether we should be treating osteoporotic patients, and patients are going off using treatments. I'm making this a long discussion, sorry. But they need to be shown, the patients need to be shown that there are actual benefits, and this is what this looks like, this is what your bone, this is normal, and this is what your bone looks like, and we've just learnt that this particular structure has an effect that we didn't know about, and it's likely that maybe you should go on treatment. And so it's actually gone back to the patient level. And we don't know if all our predictions are correct, but it certainly seems very plausible, even to the doubtful physician.
Yeah, I think that kind of, if I understood the example, it draws out an important way that models and experiments work together that maybe I wasn't emphasizing so much here. Because the cases I talked about here were mostly cases where you have some target phenomenon in the world that you're interested in, and you're building models of it, and you're doing experiments to study it, and you may be triangulating between the two to learn about it. But another really important way they work together is by building models of your experimental data, or studying the experimental system itself by building a model of it. So those are some of the things I'm thinking about with Marina O'Malley and this paper we're working on now, of when we do models and experiments in tandem, it's not always because we're modeling and experimenting to learn about a thing in the world, but sometimes we're doing an experiment to learn more about the model, and then building a model to learn more about the experiment. And yeah, that sounds like a great case of how the latter can be really revealing.
Yeah.
Just as a cynical clinician, I enjoyed some of the comments that were made before, but and I apologise for being late, but can I just draw you on one of the comments I heard when I arrived, and that was of the concerns about funding agencies and their understanding of models and experiments, and where you see how we should be approaching this topic with funders, because that has major implications for the work that universities and research organisations do. And it just worries me that with the confusion out there about the role of the various factors, that we need to be very clear when we talk to our funders about what pluses and minuses are. What's your feeling about the right approach with it?
I wish I had a better answer to that right now. I mean, sort of my big vision here is if we could change the way that people learn about science in school all the way beyond, to see that it's not the case that hypothesis testing experiments are the paradigm and best way to do science. The world would be a better place. I guess I don't have, I don't have the best sense of this stage at how to address that issue, which I think is really a communication issue and a policy issue. So far, I've just kind of been exploring the conceptual, philosophical sides of it. But I think a first step is, and I'm not sure how much this helps with funding applications, but in my mind, a first step is being really clear about exactly the sorts of things that have come out in the discussion here about how doing a model doesn't mean choosing to study an inferior stand-in. Often it means approaching a question from the most appropriate angle, given all of the background knowledge at play and given the kind of thing you want to learn about the world. So again, yeah.
As Peter's raised this issue about the different components and how to do them. One of the key things is clinical observation and raising questions and then being able to test ideas out.
Mm-hmm.
Now, if you can't attract funding to progress those things, then the potential, it doesn't matter whether it's clinical or something else, if we can't attract funding in the first place, it makes it very difficult to take it further to these levels. So it's just an interesting sort of area to sort of, where do we go? What do we do?
Yeah, so there's...
How do we actually get that message out?
Yeah, there's a related debate here, which I also worked on in my thesis, which is about hypothesis testing versus exploration, which people have talked about mainly in the context of experiments, but I think it applies equally well in the context of modeling. So that's an area where there's been a bit more discussion of the issue that you're asking about. So there's a great paper by Chris Halfie from a few years ago called Why Do Funding Agencies Prefer Hypothesis Testing, I think it's called, in Studies in History and Philosophy of Science, where I'm not sure he gives a totally satisfying answer to the how do I pitch things question, but it's just exploring these issues of why is it the case that many of our favorite stories of sexy scientific discoveries come from exploratory context, but when we go to write a paper or put things in a funding application, we have to put it in terms of the scientific method where there's a hypothesis first and then we're going to test it, and really you have to have done some of the research already before you can put it in those terms. So yeah, I think they're really deep, hard questions.
I wonder if one way out of that is to say that a hypothesis is really a model anyway. It's a model in your mind.
Mm.
A mathematical model is just a more quantitative refinement of that hypothesis. You're potentially making a prediction with a more refined tool. So it is a hypothesis. And then the iteration between the data and the model is to clearly test the model but refine it if the data requires you to refine it.
Mm-hmm.
So there's not that much difference in a way between the two.
Yeah.
Ian?
I think you're talking to the converted here, really.
Yeah.
But there was a time when basically models were paramount, and the experiment was otherwise went against the model. There was something wrong with your senses.
Mm-hmm.
And that was what, I guess 700 or 800 years ago. We've come a long way, but are there still pockets on this planet where people are, like, adhering to that view that basically, this is the way it is, or rampant raging modelers or raging experimentalists that, even in Western societies, are there still groups around like that?
Of thinking that the model is where the answers are.
You come across that, or another probably more recent example is Thomas Edison. Experiment.
Mm-hmm.
Try everything. Surely I guess that would be a very small fraction of the population now.
Mm-hmm.
You're just either in one camp or the other.
Yeah, I haven't come across someone who thinks about it today in those extreme terms, but yeah, I think there's a lot of variation, I guess, by disciplinary area and how much emphasis people want to put on trusting results from a model versus an experiment, for example. Or another thing I've been exploring in this work on surprise is sort of questions of if something goes funny, where do we assume the problem is? So often people think, if something goes funny in your experiment, at least you're learning something about the world. Whereas if something goes funny in your model, either something's wrong with your programming or you've made a mathematical assumption, but essentially it's just a bug to be fixed. So I think there's reasons to challenge that as well. Often if something goes funny in your experiment, working with microbes, for example, it's not because there's necessarily some deep thing about the question you were studying that's been answered, but just because your plates got contaminated, which I think is quite analogous to the bug in the mathematical model. So yeah, I guess that's a bit of a tangent from what you were asking about. Sorry, but I was just thinking about how when people say math is the model is the best or the experiment is the best, often what they have in mind is we can trust whatever we learn from it. And there might be reasons why that's not always the case in either context.
So it was a very interesting set of discussions, and I think we can continue it over some snacks and drinks. So let's thank Emily again for a very provoking and interesting seminar.
Thanks.